Last minute did they? You forgot your number. अभी नहीं, अभी है.
We'll start as soon as Jin comes in. We didn't expect the morning walk would be so long.
That's breakfast.
She told me that it will start at 9:50, but I just not thinking that. It will start at 9:30.
Can I use your adapter?
Which is this one? Okay.
Okay, that's your pointer.
Okay.
And there's one.
Right. Working.
Gears a little bit today, and some of you are very energized by your morning walk. And we're going to talk about Yishi Jin from UCSD is going to talk about synapse formation and signaling. And I would say that a lot of what you got in the school was about actually motor mechanics of transport. And I sort of think of problems like synapse formation as things that are related to transport may sometimes be, have central contributions from the transport processes, but transport has fundamental roles in making sure these processes work well. And Jade is going to talk about one aspect.
All right, and thank you for waiting, and I apologize for late start. So I think I come to this school with a different angle that really I'm not a trained cell biologist and nor a trained neurobiologist. And my heart is always genetics. So what I'm going to hopefully to get through is how we use genetic approaches and mutations and various approaches that's like the signaling pathways that's related to the biological process of axonal transport and synapse formation. Okay? So you'll get clear why I always love this little creature right here. And that's the C. elegans, that's with transgenic marker that's labeling neurons. But the point is, to start with, is the central question, is that we wanted to understand brain function. And what actually thinking about brain is just ensembles of many microstructures that can be identified, really can only be identified by ultrastructure. A big brain full of neuronal cells, glial cells, and vasculatures, and so on. So if you do a simple labeling, either you label axonal proteins, microtubules, and tubulins, actins, and using what nowadays is called synaptome, or neurons that essentially do a brain slice. You can stand as many different types of antibodies and co-localizations. You'll get a matchwork of molecules lining up. It could be dendrites, it could be axons, and it could be synaptic junctions, and could be any sort of structure that you're not going to be able, just by looking at immunostaining, to know where the synapses are until it's actually very classical methodology that gave us hints what the synapse look like. It's on the ultra-thin sections of brain. So this has been over 50 years now, when the EM was basically applied to biological samples, and almost the first thing they look at it, they identify is an asymmetric structure that composed of axon endings and postsynaptic cells. Okay, so, but on the other hand, this is actually a structure, a synaptic structure most of you are not familiar with, and because when you're looking at, as we're coming in, when you're looking at the synapses, you usually see around the structure with vesicles. What this is actually a longitudinal section of the axon. So within this is the axonal membrane all the way, and that's the other side of the axon membrane. So this is actually a true long axon. And within this longitudinal section, and what I really liked it is that you see the microtubules quite nicely continuous or discontinuous, as Peter Bass was showing by illustration. And also embedded in this axon, it's not very smooth structure as for illustration you often see. There's lots of bulges coming in. And within the axons, you see those are mitochondrial, right? And those tiny gray dots and the holes, so-called clear vesicles, those are the synaptic vesicles. That's components of the classical chemical transmission. And all synaptic vesicles and actually packed around a structure, is a so-called electron-dense structure, and then giving name, various people giving them as presynaptic densities or active zones, so and so. But it is the hallmark of this density and marks where the synapse site is. The asynaptic junctions and the actions of synaptic vesicle fuse with plasma membrane. Only through this action, and those are the postsynaptic cells, can be triggered a signal transduction cascade and then relay the signal in a unidirectional way, that's from a presynaptic cell and to the postsynaptic cell. And I always marvel looking at this kind of picture is that it's really a still picture that is fixed sections, ultra-thin, the animal's dead and cells dead. But this picture actually gives you a tremendous dynamics feelings. That is, along the microtubules, there are vesicles that seems to be attached to it, and you heard about motor proteins, presumably they're being transported. Okay, and you're also wondering is that then why are those synaptic vesicles and stuff here don't help onto, do they help onto the microtubules and to be carried away? So this is a typical synapses, presumably close to about 80 to 90% of the synapses and in the central nervous system are formed in a fashion is called en passant synapses. Rather than, as some of the illustrations shown, that the synapses only form at the end of axon endings. So actually synapses forms all along axons. And then basically to understand synapses formation is the central goal for over decades, many decades, is about to identify what are individual vesicles, vesicular cargos, and how do they get to where the sites are. And this is just an overview and pretty out of date, but on the other hand, mostly you can visually grasp the idea that's within the axon through the motors, and recognizing all different cargos that they will be constantly transported bidirectionally, unidirectionally, and through somewhat what they call a stochastic events, and they get stuck in a region which could be traffic jam in some ways, and that forms a so-called primitive synaptovesical release site. That is, they're not the robust synapses as what I will show you in a minute, but through those primitive sites, they can disassemble and clear the traffic jam, move on, or they can be stabilized by contact with a postsynaptic cell. By that way, and those primitive release sites gets stabilized, and through growth process by recruiting additional synaptovesicles and components, eventually becoming a mature synapse. What we define by mature synapse is actually at this point there are evoked vesicle release, and that will trigger mostly through action potential calcium influx and then trigger postsynaptic cells for vascular. But synapse is also very dynamic. Even with mature synapses, they actually undergo plasticity. They can disassemble and reform in new places. Okay. What the interesting thing about is that it's a minute biological sample structure-wise in space, but it has amazing amount of proteins. That proteomic approach has been used to study synapses in many ways, and depending on how sensitive the proteomic approaches are, it's been estimated that list over 1,000 proteins that localize to the presynaptic node, and maybe over 2,000 proteins that some are filled with postsynaptic cells, particularly in this electron-dense structure called postsynaptic density. Okay, so there's lots of proteins. And lots of those proteins also organizing vesicular structures in a very ordered fashion. This is particularly is shown that in the presynaptic terminal, which the sole function is to leading vesicle to release in a robust way. But this vesicle clustering can be physiologically and somewhat morphologically categorized into so-called reserve pools. That is, not all the vesicles are ready to release, but they are just there and to stay for additional action. And a small pool is actually undergoes vesicle fusion. It's called recycling pool. And some of the ones within the recycling pools and only small fraction contact the membrane are so-called readily released pools. That is, upon action potential, they will fuse. And eventually, this action leads to the neurotransmitter released into cleft and then captured by postsynaptic cell. Yes?
How do we specifically know that separately 1,000 are localizing on the presynaptic side and 2,000 proteins are on the postsynaptic?
How do we know this? This is all purely by biochemical fractionation. That is, you grind it up grain, and then you go through series sucrose gradient flotation, run some sort of ion gradients, and then you separate the fractions that's particularly enriched in synaptic membrane.
But we know it is pre or post through that.
The way they could do it is also through different sedimentation. So presynaptic fractions usually sedimented in one fraction more so than the postsynaptic cells, that's postsynaptic density. Generally, you're absolutely right that it's not absolutely clear, but the prep can get up to about 90% being predominantly presynaptic, 90% predominant postsynaptic. And all those biochemical purification procedures were developed in the 70s, that's through centrifugation. Okay? And, okay, so the point here is that those proteins are organized in a way for the purpose of this synaptovesical clustering. They are not only just proteins, they all have more or less a feature, so-called scaffolding proteins. In some ways, they interact extensively among themselves. One of the examples you'll see this also later is to show that the hallmark of the presynaptic terminal is the Huntington, is this little density. And the purpose of this density is so that the vesicles can be close to the density. And the density is essentially concentrated voltage-gated calcium channels. In order for that to happen, and here is enlarged view at the molecule level, that's the vesicles. Vesicles has vesicle membrane proteins that's attached to them. They were interacting with active zone proteins, as all illustrated here. You're probably not going to see that clearly, but each of the active zone proteins have also called domains, and all these arrows says that every one of them will be interacting at least with 5 proteins or more. Okay. The complexity of all of this and leading to central questions that for decades people have been trying to address. There's a lot of proteins, but what actually are the key molecules and in functioning which particular cellular process to trigger this orderly assembly and to actually localize those synapses at the right site, not just random everywhere, and what are the molecules that regulating the synaptic function, how fast release and in what way to reacting to calcium release. Okay, so if you are physiologist, what would you do to address this kind of questions? Anyone's a physiologist? Physiologists generally do is that they go through membrane potential, they try to record, they wanted to know which ion channels, which curves will be contributing to the synaptic specificity. They tend to go for the receptors, channels on the membranes, and by characterizing the channel profiles, they can guess whether it's sodium channels, potassium channels. But if you are, say, a biochemist, what would you do? I already said this. You grind it out of the brain and you try to purify proteins and then you try to see how they bind to each other. If you're a cell biologist, how do you approach this problem? Yeah, you try to tag the proteins and you wanted to see where it's going to. And the question is now, which is the key protein you want to tag? What will be the clue?
Any synaptic vesicle protein.
Any synaptic vesicle protein, okay.
In the mutants of which you see.
Right, so that's the other combination of it. You wanted to have an idea this gene, this protein is functionally important, and the only way to test that is by genetics. Okay, so that's lead to what I'm talking about is where actually I said I was not trained as a cell biologist, not trained a biochemist, not trained a neurobiologist either, and I was trained a geneticist. So the idea is that can we use genetics to identify the key molecules and from there to build the process of synaptogenesis. So I'm a geneticist working with C. elegans. I know Sandy had talked to you last week about C. elegans, a fabulous microorganism that you have to work with it through a microscope, get used to it, and it's a plate full of worm, and their life cycle is only 3 days, so it's beautiful genetic materials. And as my very first slide shows that it's transparent, and therefore you can actually see everything, and it's absolutely essential for cell biologists. The other thing is that this is the key for most of your non-genetics. You don't need C. elegans actually self-reproduce, and in a way that's hermaphrodite, you can keep genetics in a homogeneous background and really save a lot of effort in mating. And the last thing, as C. elegans people all like to do, why we love this organism, you can freeze them, and you can isolate about 100 mutations that you're not going to be overwhelmed because you just freeze them and then sell them one by one. Whoever working on flies always jealous about this, and which is one I did my PhD. Now, I don't know whether Sandy showed you this, and that is the field of C. elegans really started by this man, Sidney Brenner, when he, so anybody know what Sidney Brenner, before he became C. elegans person, what did he do? His major contribution to molecular biology? Anybody? microorganisms way after. He's the one that's helped to deduce all the amino acid codons. Okay, basically he was heart-trained molecular biologist, and by the time he came to MRC, tried working on the hardcore molecular biology. That time is when the idea is that how does amino acid encoded at the nucleotide level, and he's the one that used phage to figure out that amino acid has to be encoded by 3 nucleotides, and to figure out total together there are 20 amino acids and what codons are, and the third wobble nucleotides. Okay? So once he figured that out, he thought this is the end of molecular biology. He wanted to work on something totally different, something he was been thinking. And this is the days in the 1960s that he only need to write about 3 paragraphs and to convince the director of MRC to let him to work on something that nobody knew where he was going to. Basically, he says that really, it seems to me that he wanted to choose an organism to study the development, especially focus on the development of the nervous system. But the serious problem is actually at this stage that there's no way you define a unitary steps of any given process. And by way of saying this is that he is a reductionist. He wants to tease apart of the development process, picking each parts. So he decided that he'd like to propose what he wanted to use the approach to define each of the unitary steps is using the techniques of genetic analysis. Okay, so that genetic analysis of phage was his tool to deduce the amino acid codon. But he wanted to work on a multicellular organism with a nervous system, and he decided, so I just want to tame a small axonal organism to study development directly. Okay, and for that he decided to focus on nematodes. He studied probably over 100 species of nematode, and eventually ended up with C. elegans, because the nature of that. It's another reason is that it's the only nematode that actually can be fixed by EM and structure preserved the best, which is actually never spoken in anywhere in the history. That really is the truth. So what he did then, when he set it up, he himself is a geneticist, and he decided just getting lots of mutations. But the fact that C. elegans could be fixed by electron microscope, and then he recruited a colleague, actually at the time his technician is John White. He recruited John White to reconstruct the entire work. And that's what is the only organism we're able to reconstruct using about 8,000 ultra-thin sections of micrograph, where John White's reconstructed the whole nerve the cell bodies and the synaptic connections. Okay? So that took John White over 10 years, but nowadays, as we're looking at the brain, that there's a big push basically at the EM level reconstructing of a tiny cubic of what the brains either in the optic cortex or in olfaction cortex. Okay, so C. elegans has somatic cells is a fixed number. It's probably the only organism actually has a fixed number of cells. For a reason we still don't know why. And it's 959 somatic cells, and 302 of them are classified as neurons. John White EM reconstruction allowed him to classify them based on morphology into over 118 types, and there are estimated about 6,000 chemical synapses. Okay, so that was all the ground, but they really used the never ever go back to tell Sidney Brenner I said this, but the field don't know about it, is that Sidney used the genetics. He thought that to look for genetics of the development of the nervous system, and the best is to look for mutants that's giving behavior deficits. Makes sense, right? Brain controls behavior, and therefore you should get any mutants, any paralyzed animal that will give the genes eventually leading to the nervous system. So he collected a lot of those mutants. He named those mutants as UNC for uncoordinated. Unfortunately, the very strong UNC that he isolated all turned out to be mutants affecting muscle development. Because those animals don't move, their nervous system is actually fine. The real study of the nervous system in terms of its biology probably aided, started with Marty Chalfie, who dissected specific cell lineage that's related to the touch neurons that Samia will talk about it. I won't go into details. And the fact that Marty Chalfie later on developed the GFP as a non-invasive marker to visualize the nervous system. Now that took John White 10 years to reconstruct the nervous system, if we use a neuronal promoter to express GFP, it will take you about a week sitting in front of a microscope to know where the cells are and where they go. Okay, so the key point for going to C. elegans, a way to look at the synapses, is that there is ultrastructure conservation, despite the C. elegans and the human are so far apart. The ultrastructure conservation, as we see here. That is, there's the brain. This is what synapses you generally been taught with the textbook. A presynaptic terminal of vesicles directed to the presynaptic active zone, opposed to the postsynaptic density. That's the postsynaptic cell and mitochondria. But if you're looking at C. elegans, it is actually a neuromuscular junction, and that's a presynaptic terminal, and that's mitochondria. That's synaptic vesicles and presynaptic density, postsynaptic cell. So this level of structure is entirely conserved, and argues is that perhaps the assembly process will likely to be conserved. Okay. So with this idea going in, we decided to take the approach, combine Marty Chalfie's GFP visualization, and also the time that Sandy's postdoc advisor, Mike Noney, identified synaptic proteins as Sujita says, pick up synaptic proteins and visualize them. So we then fuse the synaptic prevalent and protein and to GFP and express them. The key here is express them in selective cells. If you express the synaptic prevalent in every neurons, as the previous slides, you're not going to be able to tell that which synaptic terminal from which apart. It will be all over the entire cord. Whereas here is we express them selectively in a subset of neurons. We call them GABA neurons, and Sandy will talk about the touch neurons. That those neurons, the soma located in the ventral nerve cord, like our spinal cord, they will extending axons to reach different part of the muscle. Is at this end, it will form this en passant synapses. It's visualized by a single puncta. Okay. Single fluorescent puncta correlating to a single presynaptic terminal identified at ultrastructure level. Okay, so now we could see, and you can track them if you're a cell biologist, just sits there and taking movies. And as geneticists, what you do, you feed that transgenic animal with mutagens, essentially nasty chemicals. And the mother is fine, and the mother will lay progenies that carry the mutation. Remember, this is only 3 days. And the other thing, remember, is self-fertilized. So therefore, we don't do any process. So that a week later, we have a brood of worm in there. A quarter of animal will have mutations, homozygous, and then you can look for what kind of defects there are. Okay. So if I were Sidney Brenner, I probably would do the first thing, pick up a bunch of UNC animals and look for whether synapses is disrupted.
You can't maintain recessive lethal mutations then?
Can I maintain the lethal mutations? Yes, you can maintain lethal mutations because the siblings are viable. So you just keep passing the heterozygous. You can look for lethal mutants. You can look for sterile animals. But if they're embryonic lethal, don't even grow to adults. You can't do it. Yeah. Okay? But this seems to be a simple. The hard work is where you do screen. So you do have to train yourself and well recognize the pattern of the... Okay. Okay, so sounds good. So you do have to train yourself and to recognize the size of the puncta and the spacing so you can recognize what is, well, now you close your eyes, okay? You guys close your eyes. Now open your eyes and then say, are those mutants or not compared to the wild type? Quite obvious, right? But remember, this is image showed it to you. But when we did the screen, is that this mutation only counts in 1 out of 1,000 animals. Essentially, on the previous slides, we do have to screen through 1,000 worms in front of us and to tell this one from a population of 100 of them. We just decided now to do behavior for a reason. We knew that behavior screens in Brenner did saturated everything, never gave a nervous system defects. It's all muscle defects. Okay, so this visual screen then led us to identify sets of mutants that are purely disrupting the synaptic morphology. Okay, now, will anybody object to this approach at this point? At least raise concerns.
It's completely GFP based.
Right, it's completely GFP based. We could completely using its artifact. It's just a GFP protein that have nothing to do with synaptic architecture. Okay, where little says, well, sure, and that's exactly the point most people say that cannot be. You know, you have to prove to us that those molecules indeed affecting synaptic architecture by localizing those proteins, or the genes and code the proteins, see where they're localized. If they turn out to be, say, cytoplasmic proteins, ubiquitins, and so on, so that could be entirely affecting the protein stability, not related to synapses, right? So, anyway, we ended up cloned each of the mutants and then found each one of them encodes a conserved protein. And then we went ahead to use a combination of factors that essentially by immunocytochemistry or GFP tagging, indeed we found every of the gene corresponding to our mutant and localized to the presynaptic terminal, as shown here. Okay, and we also did additional experiments essentially to show that the ultrastructure of the synapses is altered. Okay, so one of the examples shown here is inserted here, SID2. Is the Seattle gene SID2 encodes a conserved protein called mammalian protein called aberrant alpha. There's a worm protein, fly, and mammal. And this protein actually been identified as an adapter protein for the transmembrane receptor LAR. It's a receptor protein tyrosine phosphatase, and first actually identified by 2 hybrid to show that they bind to the intracellular domain. And this is just a GFP reporter and shows that the puncta seems to be getting diffused. Then we went ahead to do the EM ultrasection, thin section, and then reconstruct, and so that we can quantitate the number of vesicles and the length of the active zones to show that indeed that loss of SID2 actually alters the active zone size. And then for us, yes?
In the previous slide,
yeah,
you had labeled the synaptic vesicles, right, with GFP?
right.
So how do you differentiate between the synapse and the stalled vesicles?
How do I
Differentiate between the vesicles at the synapse and the stalled vesicles somewhere in the axon?
How do we distinguish? At this point, I can't distinguish it. This point, I'm relying on the fact is that, okay, so at this point, we rely on is that whatever we can detect by eye is a dense population of vesicles stuck there. If there's single synaptic vesicle transported, and my eye is not going to pick it up. And I don't care, because only those stably assembled synapses can have this level of fluorescent intensity for you to see.
Don't say you don't care. That's transport.
What's that?
Don't say you don't care.
No. I says for this screen, I don't care. And then afterwards, we did get transport mutants, but those transport mutants will have a different phenotype. That's because the transport ones got jammed. And then we start seeing them showing up in ectopic positions, is exactly what Sandy is going to talk about.
So in that mutant, when you have a lot of fluorescent intensity, that means it has so many autosomal synapses.
It appeared to be. But then when you start doing EM, you actually recognize, using additional combination factors, what defines a true synapses has to be pre and post aligned. And what defines the transport defects is usually you don't find the postsynaptic cell.
Is there a change in morphology when you see this?
Is there morphology in terms of axons or morphology? No, we don't see much of the differences in axon morphology. It's all happen within, that is the axon overall growth, outgrowth, and trajectory is all normal. So this is purely a synaptic defect. Yes?
First of all, I should say that I don't know how they did this gene. I'm still amazed because when I looked at those mutants, I can't pick them apart, and easy. So, really amazing. But the other thing is, do you get mutants in all classes? Classes in which the vesicles were spread, like you see in SID2, classes in which there were fewer. Did you get classes in which you had hyperaccumulation of cargo?
I think we got almost every class. Up to this point, I say that nobody has isolated a new mutant based on this marker that we haven't found. We found the mutants that's related to transcript biopsy, very few that punctus. Those are actually your mutants on 104 kinesin because they never got out. Okay, so you're with me up to this point? Okay, so this is just a process that as we start doing it, we never actually thought about the artifact that we just keep pursuing. In the end, synapse is a function test. Even though morphologically gets disrupted, you still wanted to know is the synaptic transmission on. So we have to do physiology, and this is where I get taught to be a physiologist, or my people get taught to be physiologists to do electrophysiology to show there is defects in synaptic transmission. Okay, and the good thing about it, since the gene is conserved, and then later on the software people show the same thing. And just to convince you, indeed, we have to accrue the data to show that SID2 is localized to the active zone. And that is, if you label synaptic vesicle, okay, and you label SID2, you can see is the density is embedded in the cluster of vesicles. It's not entirely overlapping with the vesicles because the density is only a small fraction of the synapse. And you do immuno-EM, usually the morphology is horrible, but you're able to see a couple of dots of SID2 right at the density, those are the vesicles. Yes?
One other thing, just, so what initially, so running by, back to sigma and SID2, what initially marks the site where you form, build this entire project in these neurons? What marks that this is the place the synapse is going to form?
Okay, so in this gram, we did not look for the synapse-specific genes. And that's for reason is that in this type of neuron, the entire axon is capable of forming synapses. Okay? It's still a mystery. This is still a mystery related to transport. Is that en passant synapses, there is a minimal distance to separate the synapses. What is that? Minimal distance gets controlled, and I like to ask the transport people, or the microtubule people, could that be the intrinsic factors due to cytoskeleton that needed to separate the spacing? Or related to how fast, how much of the persistivity of motor carry the cargo to go how far? That's not clear. Every type of en passant synapses, there is this minimal distance. So therefore, in the neurons we're using, we're not able to detect the specificity. But on the other hand, in the neuron that you're doing, you probably can find specificity because there is a very specific branching point to direct. Yeah. And the point is that there's more genes being showed up and models have been done. You can look for differences in specificity, assembly, transport. Naive approach leading us to the point is that we do find conserve the genes and using this approach that function at synapses. And I wanted to leave you one thing is that synapses actually is not just one form, it's multiple forms. There's incredible diversity depending on the organism, depending on the neurons. So C. elegans, like some of the mammals, have only one density with a class of vesicles, whereas Drosophila, all the insects and crayfish, and seems to have a really interesting structure called a T-bar. And that T-bar also has a lipin gene in there. And this T-bar in some ways is similar to the retinal synapses called ribbon synapses. Okay, and those are so-called CNS operated synapses. They are multiple densities scattered within one synaptic terminal. But nonetheless, all of them have a set of molecules, so-called actives of molecules, that every one of them have the same thing. They all, you can see here, is that every protein will interact with each other at least by 2 protein partners. Okay? And the idea here, a little bit running fast, is that perhaps those morphological T-bars, C. elegans, plaque, and the ribbon synapses, they have a common core of actives proteins all interacting with each other, but they can also evolve diversity through acquiring specific divergent domains. For instance, this protein is very specific for Drosophila, and that protein is actually forms this platform T-bar. Everything else is all the same between C. elegans and Drosophila, except just a little silent amino acids. There's only a certain region can form synapses to give you a quite good model to study specificity. And RPMA mutation was also identified in Sandy's old lab, and basically it's absence of synaptic patch. Okay, so essentially, when we're using genetic strings and we get identified mutations, and some other people using a different synaptic model may also identify, but possibly similar phenotype or slightly different phenotype. But the point here is that Mike's lab and my lab cloned the gene, and then realized it tells absolutely nothing because it's a large protein. It's conserved. There are human genes and there are mouse genes. None of them have been studied. And there's a Drosophila gene was studied about the same time affecting numerous junction. So with that idea, we thought, well, this has to be a very important molecule. It's conserved, and in worm affecting synapses, in fly affect the synapses, and we put our whole heart onto say, how do we figure out this protein function? The only suggestive ideas to us is the presence of ring finger domain, and that's still the early days, ring fingers then linked to protein ubiquitin mediated pathway. Okay, so I'm just going to skip. Essentially, now we did the similar thing, looking at the word argument localized, and the basic idea say that indeed argument localized to synapses. Without it, and you get a normal synapse like that. Okay? All right, so now here is the same question for you. Now, it's a novel protein, and it's a synapses affecting lucid, disrupt the synaptic morphology, therefore it's important. So you wanted to know how does it work. And if you're a biochemist, what do you do? You do Western blots, and that's give you a size. Okay, what else? Mass spec, right? So you purify the protein, you see what protein binds to it. Yeah? Okay. And anything else? Easter hybrid. Again, the idea is we're getting binding partners. And what could be the drawback of that? They may not be functionally relevant, very sticky. Okay, so we wanted to, again, using genetic approach, to see what would be the functional relevancies. Okay. Now this did take a little bit long. The reason is that when we did this screen, is we did microscopy, there's no behavior. Okay, so we can't really, unless we redo this screen using microscope and then to look for genes modify the phenotype, now being tense. So we're a little smarter, is that we start making genetic double mutants. All right, so the idea here, here's the wild type with a very nice synaptic morphology, RPM1, and give you quite abnormal synapses, but those animals actually move just fine. They're not crippled anyway. And the other gene I told you about, the SID2, also affecting synapses, and it also moves as wild type. Not much different, okay? And when we selected the double mutants, and here we go, we got an animal that's completely paralyzed, okay? And when we made this two a so-called double mutants, we got a paralyzed animal. That actually really justified the whole purpose of our screen. We hit the gold one. Is this says it's functionally the two of them are important to contribute to the animal movement, and we can correlate this paralyzed phenotype to complete loss of the synapses, both by morphology by the end. So what's good about having this synthetic phenotype is following now. We can feed the animals with mutagens, these double mutants, and then look for genetic suppressors. So that will either suppress this mutant, then you will get back a wild type. Or mutations suppress this mutant, you will also get back a wild type. Okay? So it's a very easy script. It's a paralyzed animal on the place, they don't move. And very few of them start moving, you collect them. You collect about 1,000 of them, you don't worry about it because you freeze them. And you sort them out one by one. Okay. So, okay, I'm just going to skip this one. Okay, come back to that. So we found those mutants, and then the tough job is to actually figure out their specific suppressors for the synapses. Then we go back, is from those behaviorally suppressed animals, go back looking at the synapses. Okay. And you probably ask, why do you want to look for suppressors? Because one reason is that this is, RPM1 is linked to protein ubiquitination. And that means that without this gene, whatever the other protein should be ubiquitinated will not be degraded. Okay? So the phenotype of the RPM1 is due to persistent signaling of the substrates. So therefore, by pure thinking, when knocking down the substrate of the substrate signaling, we should restore the signaling homeostasis back to normal. So that's the suppressor genetic screen idea.
But you might also get a lot of intragenic suppressors, right?
You can get intragenic suppressors. That's absolutely possible. It depends on the allele you use. So this is really critical that when you do suppressor screen, and you first need to know, are you using a non-mutation, right? If you use a non-mutation for the to revert it back to functional, it's pretty rare. If you use a missense mutation, and then you can have lock and key. And I think Sandy is going to talk about that when she's talking about transport. That definitely hits in intragenic genes. Okay? So this is the idea that we want to look for suppressors as a way to getting the substrates and the substrate to signal transduction pathway. All right. And we have a lot of them, and after we map them all, and that is still giving us many more suppressors going on 5 years work. and we find that the suppressor mutations hits 3 kinases that turn out to be a kinase cascade. So kinases are triple kinase active, a double kinase goes to kinase, and if you inactive each one of them individually, you will suppress the human phenotype.
Once you are having a suppressor, is there any way to know which gene it is suppressing? As you are having 2 double mutations in one F, so you would like to know which of the gene it is suppressing.
Yeah, so you wanted to know which gene. To be honest, this is why it was a good concept from here to here. We got tons of suppressors, and I put this as a very small letter. Just this map and cloning was 5 years from the mutation to find the gene, to establish the pathway.
Okay, this is not the right. In yours, you got a lot of hits in RPM pathway and almost nothing in the symptom pathway. So what does it actually tell you about
Yes.
the way you set up the screen?
Okay, so Cynthia leads to, this is, geneticists can constantly debate, and why in certain cases the suppressor strand works, in certain cases suppressor strand fails. And this fundamentally, that for suppressor strand, in this case, is looking for things in the absence of a gene, and it's negative regulation. Right, so a gene negatively regulates something else, then suppressor strand will work. Okay, and if you're looking for signal transduction pathways as a positive regulation, you'll be looking for things that's similar, and suppressor strand will not work. Okay, and if you're looking for proteins, protein interactions, depending on the phenotype, suppressor strand can give you compensatory changes. Therefore, you're finding the partners. And the problems why we do not get, we get very rare suppressors of liprin for reason is that assembly is a positive process, whereas a ubiquitin-mediated process is a negative regulation. All right, so we got that, getting an idea, and then some people have luck, and is that now this is early days when we try to map and clone, took 5 years to get 3 genes. On the other hand, nowadays I can have 10 mutations, whole genome sequence within 3 months. At least I have a guess what they are. Okay. All right, so I won't bore you about here. But the key point then, we really have to look for which is our substrate. Now, you have E3 ligase, you're looking for substrate, and you want it to do the first thing is that they have to be in the same domain. That is, they have to be in synapses. So to do this, we label each of the genes that we cloned, and then we see which one is the synapses. Sure enough, the upstream MAP kinases ended up localized to synapses, and sure enough, in the absence of E3 ligase, and its signal is increased, and both by immunostaining and by Western blot. And then you become a biochemist at this point. You need to show that indeed the E3 ligase combines to its a substrate, and then the E3 ligase can catalyze or stimulate ubiquitination. So with that, yes, go ahead.
In wild type, if you will mutate this suppressor, what would the phenotype?
So in this case, in the wild type, what is the phenotype of the suppressor? And it's actually superficial wild type on its own, which is why we can isolate it in the mutant, in the E3 ligase mutant, because knocking down the substrates, the animals are healthy, so therefore it can become suppressed animals for us to isolate. However, my second talk will come back to tell you that those genes, once depleted, have a new role in injury stress pathway. Okay, that is in development. Those genes needs to be kept down and through the ubiquitin process, and you completely lose it, and the organism develop actually fine. Anyway, so this is basically how, just give you an idea that we looked for suppressors, rationalize is that losing E3 ligase it will make this kinase cascade to stay high, and that's why knocking down the kinase in these ligase suppressor screens will lead you to identify the cascade. So that's all I actually wanted to, I think, say for the first part. Just thank the people who no longer with me. Mostly this work is done by a former postdoc and a former graduate student and another former postdoc. Okay, and we take a break or we ask for more questions.
More questions? Anyone?
Yes.
So what kind of suppressors are these? Are all suppressors?
This is so what kind of suppressors? They are all loss of function, full null mutations. And this all leads to why our suppressors in this case was successful, is because the E3 ligase is negatively regulating the kinase cascade. So anything knocked down, it will get me the gene. Okay. And yeah, so there are the other cases. We actually did find one suppressor mutation of a gene that what we call SID1, but not related, which turned out to be a gain of function mutation in the liver. And in that case, which is why what I alluded to is that it's very hard to find the liver pathway by suppressor screen because there is positive regulation. Yeah.
It's not directly related. Yeah, but then.
Another question I have a second question. I have a second question. Nobody else will ask me.
The student has a question.
Okay, go ahead.
So in case of injury, is RPM1 affected, then inactivated or something? In case of injury response?
In case of injury response, you say that question for my second. Okay, and then.
So when you block the ubiquitination, that is probably blocking the degradation of integrin alpha. Is that correct? Or
If I block the ubiquitination,
it's
and that will causes the kinases to be very high. So we can do the reverse experiments, is that if we just activate the kinase artificially, that will mimic the loss of E3 ligase phenotype.
why you see so many synapses. Are those synapses?
Yes, there are the synapses, yes.
The putting between alpha in that location would generate the synapse? Is that how the system works? Or why is it that when you don't integrate between alpha, you get more synapse?
Okay, so why is it that the phenotype, as if there's no synapses, right? So why is kinase activity high and leading to absence of synapses? And that's the interesting question. I don't think I have been able to answer that question. And this has something to do with synapses needs to be assembled at an optimum level. And all we could say is the outcome of this ubiquitin-mediated interaction keeps the kinase downstream of components, which my second lecture will at least give you some target genes. Their activity has to be maintained at optimum in order to have proper assembly. Right.
Any more questions? Okay, in that case, just finish ahead of time a little bit, like by 3 minutes. So we start with